Hey Bio30s. Just, reading, keeping the mind sharp. Today we are going to chat a little bit about DNA, what it is, and how to replicate it. So this is our first topic into chapter 20. Afterwards we'll then talk about how DNA is used to make proteins. So, before we talk about DNA, let's talk about, well, what is it? What does it do? DNA, also known by its full name as deoxyribonucleic acid, it's the carrier of genetic information. Now, I like to think of it as an instruction manual for you. So if you want to build something, say a protein, an enzyme, an antibody, hormone, you need instructions. Hence, we have DNA. It's able to replicate itself, permitting cell division, and of course the continuity of life. So when we replicate during cell division, we obviously also replicate our DNA. Now, history of DNA research, there's a lot that goes into, well, there has been a lot that has gone into the discovery of DNA. Mendel, who we'll talk a ton about in the genetics unit, he showed that DNA, or traits, right? Traits that are sections of DNA on our chromosomes can be passed from parents to kids. Morgan showed that genes are located on chromosomes. So a gene being a long segment of DNA, they're located on our chromosomes. So technically our chromosomes being the units of inheritance when it comes to our traits. Chargaff showed that the amount of adenine will always be equal to the amount of thymine, which are two base pairs within the DNA molecule. That also goes for cytosine and guanine as well. There we go. Rosalind Franklin, she was one of the first people to actually see an X-ray picture of a DNA molecule. She noticed that the double helix shape of that molecule, we can see the inside nitrogen bases here and the outer sugar phosphate backbone. And then her research was also built upon by Watson and Crick. So they proposed a 3D model of DNA back in 1953 showing the twisted double helix staircase that we know today. Now, unfortunately, Rosalind Franklin didn't really get much credit at that time. It's mostly Watson and Crick that got the credit. She did later get the credit that she rightly deserved, but unfortunately she wasn't around to receive any awards because she had passed away due to complications from the very X-rays she was using to look at the DNA. So when we talk about DNA, like I mentioned, it has that twisted double helix structure. So it looks like a ladder that someone has just twisted. The backbone of that ladder is made of sugars and phosphates. So phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. The inside, or the inner rungs, are made of nitrogen bases, and they can be one of four nitrogen bases. They can be guanine, cytosine, adenine, or thymine. And then those nitrogen bases are stuck together with hydrogen bonds. I won't go into the chemistry of hydrogen bonds, but if you've taken Chem 20 or 30, you should know what they are. Generally, they're weak by themselves, but when you couple multiple hydrogen bonds, as well as that twisted nature, a DNA molecule is actually pretty secure when it comes to the two sides being bonded together. Now, when we have those nitrogen bases fitting together, we have a pairing. So I like to think of a good way to remember this pairing. Oops, let's jump back there. So a good way to remember this pairing, I like to use the acronym GCAT. Okay, so G, guanine, always pairs to C, cytosine. So a purine, like guanine, will always pair with a pyrimidine, known as cytosine. And then adenine, which is also a purine, will bind with thymine, which is a pyrimidine. So those two will always stick together. So if you see a G, there's a C attached to it on the other side. If you see a T, there's an A attached to it on the other side. And then leading into Chargaff's rule one more time, the amount of A will always equal the amount of T, the amount of G will always equal the amount of C. So for example, if we say there's 20% of the molecule is thymine, that means 20% of the molecule is then adenine. So 20 and 20. That would mean there's 30% guanine and 30% cytosine. So generally speaking, A always equals T, G always equals C in terms of percentages. All right, so here's the whole big DNA molecule put together here. We have our phosphate sugar backbone, our nitrogenous bases in the middle. We also have what's called nucleotides. Now what a nucleotide is it's essentially one phosphate, one sugar, and one nitrogenous base. That's a nucleotide. There's another nucleotide, there's another nucleotide, and there's another nucleotide. And then we have nucleotides on the other side of the molecule. Now, excuse me, when we run down the DNA molecule, you'll notice that this end right here is a 5' phosphate running all the way to a 3' sugar, okay? On the other side, you'll notice that these are they're upside down. It's because DNA runs anti-parallel to one another. So we run 5' to 3' in this direction, and then over here, our 5' is down here, our 3' is up here, so we run in the opposite direction. So DNA is anti-parallel to one another. In terms of the primes, the 3 and the 5, all that really means is what carbon the molecule is bound to. So if you've taken Chem 30, you might know about organic chemistry, labeling carbons. All this means is the phosphate is attached to carbon number 5 in the sugar. Okay? All this means is the OH group, there should be an OH group at the bottom here. The OH group is attached to carbon number 3. So here, phosphate group attached to carbon number 5. Over here, OH group attached to carbon number 5. Oh, sorry, carbon number 3. It should be carbon number 3. I didn't draw that very well. So this should be your 3'. Okay, so that's the basic structure of DNA. Now that we know the structure of DNA, we need to get into how does DNA replicate itself. Now remember, DNA replicates itself during the S phase of the cell cycle. This is where a single-stranded chromosome will now become a duplicated double-stranded chromosome. DNA replication is what we call semi-conservative. So what the DNA will do is actually unzip itself. So we actually take a pair of scissors or like unzipping a zipper, and we break apart the two strands of DNA. And we use those strands as a template to fill in the missing pieces to make two brand new strands of DNA. But these new strands of DNA are identical to the original strand of DNA. So it's a long process involving multiple enzymes, unzipping, adding, and then of course proofreading to make sure that we've done a good job, because any errors in DNA replication will show through all of the cells that divide afterwards. So if then we go through mitosis, that error is present in the two new cells, and then those cells divide, that error is still present, and those errors are mutations, right? So we'll chat a little bit about mutations later. So how does this process work? Well, step one is we have to unzip the DNA. How do we unzip the DNA? Well, there's a special enzyme. This is one of 4 enzymes that I need you to know. The first enzyme is called DNA helicase. And what DNA helicase does is it unzips or breaks apart the bonds, the hydrogen bonds between the nitrogenous bases. So the point at which the two strands are separating, we call the replication fork. So right here, the replication fork would be here, and the direction of replication would be this way. We're unzipping this way. So basically, helicase attaches itself to DNA and then just starts breaking apart hydrogen bonds. So it just starts unzipping those hydrogen bonds. And it keeps unzipping and unzipping and unzipping, giving us 2 strands. Now we need to fill in the missing pieces. One strand, or one template, runs 5 to 3. The other will run opposite 3 to 5. And this is really important because when we talk about the next enzyme, direction matters. So the next enzyme we're talking about is DNA polymerase. What is DNA polymerase doing? Well, DNA polymerase is taking free nucleotides. Remember, a nucleotide is a phosphate, sugar, and a nitrogenous base. So it's taking free nucleotides and sticking them to the corresponding nucleotide. So if you have a G, polymerase is going to take a C. If you have an A, polymerase is going to take a T. Where do these nucleotides come from? They come from the food you eat. So you actually get nucleic acids or building blocks of DNA from the foods we eat. So there's two types of DNA polymerase that we're going to talk about. There's DNA polymerase 3 and there's DNA polymerase 1. So here's what DNA polymerase 1 does. DNA polymerase 1 is kind of your proofreader, and it will kind of cut out what are called RNA primers, and I'll explain the RNA primers in a second. So they will cut out the RNA primers and replace them with the appropriate DNA. DNA polymerase 3 is the one that's doing most of the heavy lifting. So polymerase 3 is the one adding those free nucleotides to the top strand and the bottom strand. One strand is running 5 to 3, which we call the leading strand, and the other strand is going in the opposite direction, 3 to 5, which we call the lagging strand. The leading strand is really easy to replicate. It just goes boom, 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 one continuous stream. The lagging strand, unfortunately, DNA polymerase 3 can only copy in the 5 to 3 direction. So if you have to go backwards 3 to 5, you actually have to work your way backwards, making the DNA in short chunks or fragments called Okazaki fragments. Then we cut out the primers, replace them with DNA. We get our fourth enzyme, DNA ligase, gluing it all together, and we're done. So, clear as mud? Probably not. So let's go through one more run through of DNA replication here. I'll go through it with a diagram, okay? Step 1, DNA helicase unzips the DNA. So DNA helicase unzips the DNA. We're replicating in this direction. So helicase unzipping our DNA strand, breaking apart those hydrogen bonds, okay? Now, I mentioned I was going to talk about primers. What a primer is, it's like a place for the enzyme to hook onto. It's a starting point for the enzyme. So we're going to place an RNA primer right here, okay? So now, DNA polymerase 3 is going to hook onto this primer and start copying the leading strand in one continuous motion. Because you'll notice we're going 5 to 3. 5 to 3. So polymerase 3 can go 5 to 3. So it just needs one primer to start, and then it just builds that DNA strand in one continuous motion. Let's look at the lagging strand. Notice how the lagging strand is going 3 to 5. So we can't go that way. The polymerase can't go 3 to 5. It needs to replicate this way, 5 to 3. So we got to know that we're going 5 to 3, not 3 to 5. Now, how does this work? Well, to do this, we have to lay down multiple primers. So we have to lay down multiple hooks that the DNA polymerase can hook onto, copy, 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 hook onto, copy, 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 hook onto, copy, copy, copy. The best way I can explain this, and it's a little bit of a weird analogy, but this is the best way I can explain this, is you're running a race, okay? So one person is running the race, they go at the starting line, and they run, okay? Start to finish. That's your leading strand. Start to finish, easy. The lagging strand is, the person is running a race, but they hop in a golf cart, they take them a quarter of the way up, and then they run backwards to the starting line. Then they hop back in the golf cart, takes them up to the next point, the middle of the race, and then they run back to the quarter point, and then the 3/4 point, and then to the end. And they're just running backwards. Are they covering the same distance? Yes, they are. Are they doing it in a strange roundabout way? Of course. But that's what's happening with the lagging strand. We can't do it in the direction we want to do, so we have to do it in short fragments called Okazaki fragments, named after the scientists who discovered these fragments. So we'll make one fragment, another fragment, another fragment. We're still making a strand of DNA. We just have to work backwards. Okay? So now, here's where we're at. We have the lagging strand done. We have the leading strand done. We just have to kind of put some finishing touches on there. So we're going to get DNA polymerase 1. It's going to come in, and it's going to remove these primers. Remember, RNA, DNA, they sound the same, but they're not the same. They're slightly different. we can't use RNA when we're making a strand of DNA. So we're going to remove our attachment points, remove the primers, and replace them with the appropriate DNA molecules. So we're going to remove these primers and replace them with DNA. That's the job of polymerase 1. Now, both polymerase 3 and polymerase 1 can proofread the DNA. So they might do a quick once-over, proofread the DNA to make sure there's no mistakes. It's as if it's the same thing as you would never hand in an essay to your English teachers or to your social teachers without proofreading it, or at least I hope you wouldn't. Or you wouldn't hand in an exam without at least going through the questions first. So you proofread to make sure you've done a good job, and then finally the last enzyme, DNA ligase, comes in and glues those fragments together. Because when we make fragments, there's small gaps in between. We don't want gaps in our DNA. So we glue the fragments together, ligase glues it all together, and now what we've done is we've used one strand of DNA, unzipped it, and then used that as a template to make 2 strands. These 2 new strands, however, are exactly the same as the original strand. So now we have a double-stranded or duplicated chromosome. We've copied all the DNA in that chromosome, and now we're ready to move forward with either mitosis or meiosis. So that's it for today. Next lesson, we're going to get into the process of how we read DNA and then convert that into a building of a protein. So we go from the transcription and translation, so going from DNA, amino acids, all the way to a protein. So thank you for watching, and we'll chat again soon.